Welcome to this lecture on the biochemistry of metabolic processes. Today, we are going to explore the fascinating topic of gluconeogenesis. This is a truly remarkable and life-saving metabolic pathway that your body uses to manufacture glucose from scratch when you are fasting, starving, or exercising intensely. Let us dive in and see exactly how it works. To understand why this pathway matters, imagine you are on a long road trip in a car that only runs on a very specific premium fuel. Suddenly, your tank is empty. If you had a magical backup generator that could take random scraps from the trunk and convert them into that exact premium fuel, you would be saved. In your body, gluconeogenesis is exactly that magical backup generator. It takes metabolic scraps and turns them into fresh, life-saving glucose when your stored reserves run out. Now that we have our analogy, let us look at the biological reality. Why do we need this backup generator? Your brain is a massive energy consumer, requiring about 120 grams of glucose every single day. Unlike your muscles, it cannot easily switch to burning fats. Furthermore, your red blood cells completely lack mitochondria, meaning they are absolutely dependent on glucose to survive. If gluconeogenesis did not exist, your blood sugar levels would crash to zero within a day of fasting, leading to a coma and death. So, what exactly is this life-saving process? Formally, gluconeogenesis is the metabolic pathway through which organisms synthesize new glucose molecules from non-carbohydrate precursors. The word itself breaks down beautifully. Gluco means sugar, neo means new, and genesis means creation. Instead of breaking down carbohydrates from your diet, your body takes alternative molecules like lactate, glycerol from fats, and certain amino acids to build glucose from the ground up. This heavy lifting takes place primarily in the liver, with a small amount occurring in the kidneys. You might be wondering, is this just glycolysis running in reverse? The answer is no. Gluconeogenesis is not simply the exact reverse of glycolysis. Glycolysis has three highly irreversible steps that act like one-way turnstiles in a subway station. You cannot just walk backwards through them. To solve this, gluconeogenesis uses special bypass enzymes to navigate around these one-way roadblocks. This clever detour ensures that the metabolic pathway flows strictly in the direction of glucose production, even when the cell is trying to conserve energy. Now that we understand the pathway's purpose, let us look at its energetic cost. Building a complex molecule like glucose from smaller pieces is an uphill battle. Here is the overall balanced chemical equation. We start with 2 molecules of pyruvate, a 3-carbon molecule, to build one 6-carbon glucose. To drive this reaction, the liver must invest cellular energy in the form of ATP and GTP, along with reducing power from NADH. If the availability of ATP decreases in the liver, this entire pathway slows down because the energetic toll is simply too high to pay. Let us break down exactly what this equation costs the cell. We want to find the total number of high-energy phosphate bonds required to make just one molecule of glucose. First, we look at the left side of our equation and see that 4 molecules of ATP are consumed. Next, we see that 2 molecules of GTP are also consumed. Since GTP is energetically equivalent to ATP, we simply add them together. 4 plus 2 gives us 6. So making one glucose molecule costs a massive total of 6 high-energy phosphate bonds. This high cost leads us to an important calculation, the net energy deficit. We need to compare the energy spent building glucose via gluconeogenesis with the energy gained by breaking it down via glycolysis. The formula is e-Net equals the sum of e-ATP and e-GTP minus e-gain. e-ATP and e-GTP represent the energy spent, while e-gain represents the net ATP produced during glycolysis. By calculating this, we can find out exactly how much energy the body loses if it were to run both pathways in a complete cycle. Let us calculate this net energy deficit using our known values. We know that gluconeogenesis uses 4 ATP and 2 GTP molecules. We also know from glycolysis that breaking down one glucose yields a net of exactly 2 ATP. We substitute these values into our equation. 4 plus 2 equals 6, which is our total cost. We then subtract the 2 ATP gained. 6 minus 2 equals 4. The final answer is a net loss of 4 ATP equivalents per cycle. This is a significant energy drain on the cell. What does this net loss of 4 ATP actually mean for the body? It explains why the body tightly regulates these pathways. If glycolysis and gluconeogenesis ran at the exact same time in the same cell, it would create a futile cycle. The cell would rapidly burn through its ATP reserves, generating nothing but heat and wasting precious cellular energy. To prevent this disastrous waste, hormones like insulin and glucagon act as master switches, ensuring that these two opposing pathways never run simultaneously. Now that we understand the regulation, let us look at a real-world scenario: heavy exercise. When muscles work anaerobically, they produce lactate. The liver converts this lactate back into glucose. To find out how much glucose is made, we use this simple formula. N-glucose equals N-lactate divided by 2. N-lactate is the starting number of 3 carbon lactate molecules. We divide by 2 because it takes exactly 2 of these 3 carbon halves to make one 6 carbon whole glucose molecule. Let us see this formula in action. Imagine an athlete is sprinting intensely, and their muscles produce 10 molecules of lactate as a byproduct. This lactate travels through the bloodstream to the liver. We want to know how much glucose the liver can produce. We take our starting value, N-lactate equals 10, and plug it into our equation. 10 divided by 2 equals 5. The final answer is that the liver will produce exactly 5 molecules of glucose. All 20 carbon atoms are perfectly conserved during this conversion. We know how much glucose is made, but what is the energetic price tag for the liver? To find the total energy cost for this conversion, we use this formula. C total equals N glucose times 6. C total represents the overall energy burden on the liver, measured in ATP equivalents. We simply take the number of glucose molecules produced and multiply it by 6, which is the known energetic cost we established earlier for building a single glucose molecule. This scales the cost up to the total batch size. Let us calculate the final energetic price tag for our sprinting athlete. We determined in the previous step that the liver is producing 5 molecules of glucose. We substitute N-glucose equals 5 into our total cost equation. We multiply 5 by the cost of 6 ATP equivalents per molecule. 5 times 6 equals 30. The final answer is that the liver must spend a massive 30 ATP equivalents to perform this life-saving conversion. This is a heavy burden, paid for by burning fatty acids in the liver. This entire scenario we just calculated perfectly describes a vital physiological process known as the Cori cycle. In this cycle, the working muscles get to dump their metabolic waste, lactate, into the bloodstream. The liver acts as a generous donor, taking in this waste and taking on the heavy energetic burden of 30 ATP equivalents to recycle it. The liver then pumps fresh glucose back into the blood for the muscles to use. This beautiful teamwork allows your muscles to continue contracting during intense physical exertion. Let us visualize how this pathway actually navigates around the roadblocks of glycolysis. On the screen, you see a flowchart comparing the two pathways. Glycolysis flows downward, while gluconeogenesis flows upward. Notice the 4 enzymes highlighted in green on the right side: pyruvate carboxylase, PEP carboxykinase, fructose 1,6-bisphosphatase, and glucose 6-phosphatase. These are the 4 unique bypass enzymes we discussed earlier. They act as detours, allowing the liver to push the reactions upward against the energetic gradient, ultimately synthesizing fresh glucose at the top. We have covered a lot of ground today, so let us review the key takeaways. Gluconeogenesis is the vital pathway that synthesizes new glucose from non-carbohydrate precursors, like lactate and amino acids. It is not just reverse glycolysis. It relies on 4 unique bypass enzymes to overcome irreversible roadblocks. This process is incredibly expensive, costing the liver 6 high-energy phosphate bonds per glucose molecule. Ultimately, this energetic sacrifice is what keeps your blood sugar stable and ensures your brain and red blood cells survive during fasting. Great job following along.